In the quiet moments after someone asks a question like this, there is often something deeper beneath it. A mix of curiosity, fear, biology, and myth. Can sperm survive in water? It sounds simple, almost casual. Yet it opens a door into one of the most misunderstood corners of reproductive science. Many imagine water as a gentle environment, neutral, harmless, maybe even protective. A place where sperm could float, pause, or wait. But the truth is far more precise. Sperm are engineered for one purpose and one environment: the human reproductive tract. They depend on tightly controlled pH, exact temperature, and biochemical cues crafted for their survival. Drop them into water, and everything changes. Their delicate membranes begin to strain. Their internal chemistry destabilizes. Osmotic pressure pulls at them, stretching and collapsing their structure. Within seconds, the microscopic engines that drive movement and protect DNA begin to fail. But the twist is this: not all water is the same. Fresh water destroys sperm through swelling. Salt water dehydrates them. Chlorine breaks down their membranes. Hot tubs accelerate their collapse. Even bath water, seemingly harmless, dilutes and disables them long before any survival matters. Rumors, fear, and what-if scenarios have confused generations. But the real science is clearer than the myths. To understand why sperm behave the way they do, we must examine their anatomy, their fragile chemistry, and the hostile environments that water creates the moment they leave the body. This is not just a story about survival. It is a story about design, precision, and the limits of cellular life. Let's go inside. To understand what truly happens when sperm meet water, we have to zoom inward, beyond the myth, beyond the what-if, into the microscopic architecture of a cell designed with extreme precision. Sperm are not built for versatility. They are built for one journey, in one environment, following cues that exist only inside the reproductive tract. A sperm cell is composed of three fragile parts: the head, holding tightly packed DNA; the midpiece, where mitochondria generate energy; and the flagellum, the tail responsible for propulsion. Each structure relies on exact chemistry. Even slight environmental changes disrupt the system. Inside the body, sperm move in a protected medium, seminal plasma, rich in sugars, proteins, buffers, and ions. It maintains pH, shields their membranes, and fuels their movement. But the moment sperm enter water, this protective layer dilutes instantly. Without it, sperm face the environment directly, and that environment is fundamentally hostile. Fresh water has a solute concentration far lower than bodily fluids. Osmosis immediately takes over. Water rushes into the sperm cell, swelling it like a balloon. The membrane, made of delicate phospholipids, cannot tolerate the sudden pressure. It stretches, weakens, and in many cases ruptures within seconds. Even if the cell stays intact briefly, its ability to move collapses nearly instantly. Salt water works in the opposite direction. With a higher solute concentration than the body, it pulls water out of the sperm. The cell shrivels, enzymes misfold, and the flagellum stiffens. Movement becomes impossible long before structural collapse occurs. Chlorinated pool water is even harsher. Chlorine exists to break down organic matter. To a sperm cell, chlorine is direct chemical aggression. It denatures proteins, destabilizes DNA, and disrupts the mitochondrial engine. In most cases, survival is measured in seconds. Hot tubs and warm baths create thermal stress. Sperm depend on stable, cooler temperatures during development, and rapid heat exposure accelerates their metabolic breakdown. Enzymes lose structure, mitochondrial energy is depleted, and the tail loses flexibility. Cold water slows biochemical reactions to near zero speed. Even if the membrane stays intact briefly, movement becomes biologically impossible. Without movement, sperm cannot fulfill their purpose. Yet chemistry alone is not the whole story. Even if water did not damage sperm, dilution would. For sperm to function, they must exist in extremely high concentration, tens of millions moving in the same direction. Water scatters them immediately, diluting their power to a level that renders them biologically irrelevant. Direction disappears. Chemical signals disappear. Energy sources disperse. The chain required for their mission collapses from the first moment. Inside the reproductive tract, sperm rely on cues, pH gradients, glucose availability, cervical mucus pathways to guide them. None of these exist in water. Instead, the tail flickers weakly, sometimes once or twice, before the internal machinery shuts down. In bathwater, the most common source of fear and misconception, the story is the same. Even if a few cells are momentarily intact, they cannot move with purpose. They cannot withstand osmotic change. They cannot survive without concentration or direction. By the time water dilutes the seminal fluid, function is lost. What remains is not a sperm ready to act, but a cell unprotected, scattered, and chemically collapsing. The deeper we look, the clearer the story becomes. Sperm are exquisitely engineered, but only for a narrow environment. Water unravels that engineering almost immediately. The biology leaves little room for myth and no room for misunderstanding. In the end, the question, can sperm survive in water, reveals something far more profound than a simple yes or no. It exposes the extraordinary precision of human biology, how cells are engineered not for versatility, but for purpose. Sperm are not generalists. They do not adapt to different environments, wait in suspension, or pause their mission for later. Their survival depends on a narrow window of chemistry, temperature, and concentration that exists almost exclusively inside the reproductive tract. Water, in any form, breaks that window open. Fresh water overwhelms them. Salt water dehydrates them. Chlorine dismantles their structure. Heat accelerates their collapse. Dilution dissolves their direction and purpose. And yet, these vulnerabilities reveal the deeper beauty of biological design. Reproductive cells are meant to function only where life can begin. They are not built to drift through accidental environments. They are built to respond to cues, gradients, and protective systems that guide them toward a single destination. For many, understanding this science brings relief, a release from myths, fears, or questions left unanswered for too long. For others, it illuminates how delicate and specialized the beginning of life truly is. Reproduction is not random. It is not easily triggered. It requires an extraordinary alignment of conditions. Sperm in water are not agents of possibility. They are cells out of context. Cells without their signals, without their medium, without their purpose. Their quick breakdown is not failure. It is biology doing exactly what it is designed to do. Water is where sperm lose their structure, their direction, and their meaning. And in understanding this, we gain a clearer sense of how life protects its origins, through precision, through limitation, and through environments that must be just right.